Welcome everyone. My name is Dr. Carl Giordano. I'm a medical doctor and I created this lecture to educate and update you on our current understanding of lifespan, health span, nutrition, and fitness, and how at any age you can positively impact these categories. This is not just about living longer, but about living healthier. After you listen to this 30-minute lecture, you will understand the biology of this field, and once you do, I think you'll find this information valuable and compelling. This is not new information. This is information that was constantly advanced over the last 15 to 20 years. I put this lecture together because it's my feeling that this information is just too valuable for the public to not fully understand. This opening slide is not some random picture of a tree. This is a picture of a bristlecone pine tree, the longest living organism on the planet. This tree has segments of its DNA identical to our human DNA. These segments of DNA that promote longevity are called longevity genes. This organism simply has more of these genes than we do. We are going to spend a lot of time in this lecture talking about the biology of these genes and how they impact our lifespan and health span and how you can benefit from this knowledge. Although we talk about these topics individually, I want this picture to capture the fact that these all overlap. Lifespan, health span, exercise, and food. I want to emphasize that this is not just about living longer. This is about living healthier and more athletic. I'm not talking about food or exercise, and I'm hoping to post another lecture on those categories soon. Now, when we talk about influencing the aging process, I'm talking about extending life, reversing aspects of the aging process, and living a healthier, more athletic life. People find this unbelievable, and I hope this lecture explains to you why this should be very believable. These two gentlemen received the Nobel Prize back in 2012 for their contribution to the field of cell programming and their work on extending our understanding of cell aging, cell identity, and age reversal. Gurdon, pictured to the right, just to give you an idea, back in 1962, showed that cell specialization was reversible. Yamanaka, back in 2006, pictured on the left, showed that it was possible to reprogram an adult cell to reverse its age all the way back to a stem cell. And continued research done around the world over the last 15 years has continued to extend our understanding of this science. So this process is anything but science fiction. This information just doesn't come across your desk. But I think this information is so valuable that somebody like myself has to try and get this information into the public domain. Now, Sinclair from Harvard has focused on the idea that aging is the disease, and I think he is spot on. Underneath this umbrella of aging are all the health conditions that you hear about as people get older. This will become clear as my presentation proceeds. So yes, we're going to talk about the small things in the cell, and we will review some basic science today, and I apologize in advance for that. But we need to see the small things in the cell in order to understand the big ideas. So the next 20 slides or so are essentially a review of some basic biology and genetics to provide a foundation so you can better understand what we're going to be talking about. And then we're going to talk about some very specific messenger molecules that can nudge our cellular functions in our favor to promote longevity and a more healthy athletic life. So the picture on the left is Darwin's 1837 handwritten idea of the tree of life and evolution. The picture to the right is our modern tree of life that we accept, and we now have DNA sequencing that allows us to analyze the genetic relationships between species and confirm that there are genes throughout this tree of life that we all share. And through evolution, more complex organisms developed, namely humans. We're obviously over here in the eukaryote domain. We're going to focus on 3 specific genes that are, again, conserved throughout evolution and throughout this tree of life and are amenable to manipulation. This tree of life is the reason why we can experiment on different organisms, and what we learn from them we can apply to humans. So we're going to focus on 5 major molecules found in nature, and we will go through them one by one. These 5 molecules have been studied for decades, some even longer, and all of these have hundreds of publications supporting their benefits, their tolerance, as well as the benefits of extending lifespan and improving health span and providing a more athletic life. All of these benefits appear to be true when adopted at almost any age. Okay, so let's just review some basic science so you can understand how these signal molecules may work. Inside the nucleus of a cell is the chromosomes, and the chromosomes are made up of your wound DNA and sections of the DNA that code for a specific protein. These sections are referred to as genes, and we're going to focus on 3 specific genes. In reality, you have about 20 to 25,000 genes and 23 pairs of chromosomes, but again, in this lecture, we're going to be focusing on just 3 specific genes. So there are 3 cellular processes that we need to be aware of. I'm just going to review them. The first is protein synthesis. Recognize every cell in your body in the nucleus has the exact same DNA. What determines a brain cell from a skin cell is what proteins that cell makes. The intracellular proteins give the cell its identity. So think of the DNA as a zipper, and the DNA unzips, and through a process of transcription, it makes RNA. The RNA then leaves the cell, attaches to a ribosome, and makes proteins. Some of these proteins stay in the cell and determine the cell identity. Some of these proteins exit the cell and have extracellular functions. For example, elastin, collagen, and hyaluronic acid leave the cell and keep your skin moist and plump. Keratin leaves the cell and provides the material for your skin and your fingernails and your hair. So the more perfect this process, the longer your cell maintains its identity and specialization, and essentially the slower you age. The more imperfect this process, the faster your cell loses its identity and specialization, and the faster you age. So these proteins that stay in the cell are the proteins that determine the cell's identity. The second cellular process is mitosis. Some cells undergo a lot of mitosis, like skin cells. Some cells undergo very little mitosis, like your brain cells. But a mother cell basically makes an exact replica daughter cell. If this is a skin cell we're talking about, failure of this process contributes to wrinkles. This is part of the process of how we grow and how we maintain ourselves. And again, the more perfect this process, the slower you age. The third cellular process that you need to be aware of is something called autophagy. Autophagy is the recycling of proteins inside the cell. Sometimes these cells are made, but they are misfolded in the wrong 3-dimensional configuration. And when that occurs, they don't provide any function inside the cell. But we want to recycle them, because if they remain in the cell, they create cellular clutter, and they decrease the efficiency of the cell. So these are the 3 basic processes that you need to be aware of. And everything we're going to talk about today is keeping these processes working efficiently and accurately. Aging basically starts to occur when these 3 cellular processes start to malfunction. Okay, a little bit more background. There are 2 types of data in biology: the genome, which is your complete set of genetic instructions, and the epigenome, which is a multitude of chemical compounds that turns your genes on and off. So this is a pictorial representation of DNA, the twisted ladder or double helix. This is a more organic chemistry representation of DNA. And the current thinking is that changes in the genome is not the reason why we age. Your genome remains relatively stable, which is why we can take any cell in your body and clone a brand new you. So the original information from your genome remains. The epigenome describes a network of chemical compounds that modify the epigenome without altering the DNA. It does this by spooling and unspooling the DNA around balls or histones. This is basically how your body turns your DNA sections on and off. When the DNA is wound up around the histones, it's essentially not allowing that section of the DNA to go through transcription and translation and make proteins. This is how your body creates a brain cell or a skin cell, by essentially turning certain sections of the DNA on and off. So cells maintain their identity as long as the correct on-off switch network exists. As this on-off switch network gets corrupted, your cells lose their identity and you start to age. So it's not the degradation of the DNA, but the degradation of the spooling and unspooling of the DNA that leads to aging. So this is important. This is what you have to understand. And we'll talk a little bit more about this in the next few slides. So this spooling and unspooling is what determines your cell identity, and it determines the cell's ability to main itself through protein synthesis. So sections are unspooled, and then this transcription, translation, and protein synthesis pathway is underway. So this winding and unwinding of DNA is part of a highly coordinated ballet that the cells go through to stay healthy and specialized. And we refer to this as an epigenetic dance. Imagine the wrong genes turned on and off in a brain, in a brain cell. All of a sudden, the brain cell starts to take on characteristics of a skin cell, and that is not going to allow the brain to function normally. If you want to promote health span and lifespan, you want to keep your cells maintaining their identity by keeping the cells spooled and unspooled in the correct fashion. Don't forget, your cells make up your tissues, and your tissues make up your organs. So corruption of this spooling and unspooling process can lead to accelerated aging. So recognize every cell in your body, again, has the same DNA, but different cells have different genes turned on and off by spooling and unspooling these areas. We need this process to remain accurate because as this process degrades, this is the origin of aging. So corruption of the epigenetic information, corruption of the spooling and the unspooling is what leads to altered mitosis and altered protein synthesis, which lead to aging. So Sinclair from Harvard refers to the inability to maintain cell identity as X differentiation. We want our cells to stay fully differentiated or specialized. We want our brain cell working as a brain cell. We want our muscle cell working as a muscle cell. We don't want our brain or muscle cell working as a skin cell. Because when they do, we refer to them as being X differentiated. Okay, so just a little bit more on how the genome works. This spooling and unspooling process occurs this a little bit and refer to this as a methylation reaction as a way to promote the spooling and unspooling. A methyl group is simply added to the nitrogen base. These hairs that you see, these histone hairs, will either hold the DNA tightly packed or release the DNA and allow it to be unraveled so it can go through transcription and translation. This is how the cells turn genes on and off. This is how your epigenome is influenced. This is how your cell determines its identity and maintains the health and specialization of your cells by turning genes on and off. If you want to age faster, you corrupt this information. This is essentially the information theory of aging. Okay, so that's how the epigenome works. I'm going to now focus on 3 specific sections of DNA or 3 specific genes that make the Sirtuin protein, the AMPK protein, or the mTOR protein. And we're going to focus on those because those appear to be the most important and those are supposed to be the most amenable to manipulation. And I apologize for that science lesson, but I did say in order for us to talk about fine-tuning in biology, we have to go into the small things into the cell. So we're now going to talk about those 5 molecules that can influence these 3 longevity genes in our favor. And we're going to start with Sirtuins. And there are hundreds of longevity genes, but these 3 appear to be the most important. And these, again, are highly conserved evolutionary genes found in all forms of human life, unicellular organisms, plants, elephants, and humans, and everything. Okay, so Sirtuins are a family of signaling proteins or enzymes involved in metabolic regulation. They are, again, highly conserved through evolution and are found in all forms of living organisms. So these enzymes are the enzymes that add or remove that methyl group from the histones that turn the genes on and off, and they are referred to as guardians of the genome. By doing this, they control what proteins our cells make and they determine our cell identity. You can think of these sirtuins as workhorses inside the cell that are constantly moving along the genome, controlling the spooling and unspooling process. These proteins also participate in a dual function, not just to guard the genes, but also to repair genes. So if you remember anything from this lecture, you need to remember how important the sirtuins are. I want to just touch on this dual function system again. Not only do the sirtuins maintain cell identity by controlling this gene switch network, they also participate in repair of DNA. So when DNA is damaged, the sirtuin has to migrate away to repair that DNA. And the type of DNA damage that's most consistently linked to aging occurs at a rate of 10 to 50 times per cell per day. So the more damage that occurs in your DNA, the more the sirtuins have to migrate away from guarding their specific genes. And sometimes these sirtuins can't migrate back to their post. And now you can begin to see the domino effect. If the sirtuins can't get back to where it was, certain genes may start to be turned on and off incorrectly. So as sirtuin function decreases, our tissues start to age because of these epigenetic changes. We lose our cell identity. This is seen in all tissues: the brain, the heart, the muscle, the kidneys, the eyes, the skin, the hair, everything. And things that decrease sirtuin function are what you can imagine: increased calorie intake, obesity, increased sugar, blood sugar, smoking, alcohol, radiation damage, sun exposure. All those types of things will decrease sirtuin function. So now we can start to focus on these 5 major molecules and how these molecules may work in our favor with these longevity genes. These 5 molecules get a tremendous amount of press today. They've all been studied for years, and published results exist on all of them. These 5 molecules provide the benefits that we are all seeking, namely a long and active and healthy life by working at the level of the epigenome to help maintain our cellular identity. So we'll start with this first one. So again, without the sirtuins, the epigenome is compromised and a domino effect occurs. Protein synthesis becomes uncoordinated. Cell replication through mitosis is compromised. All the hallmarks of aging. And if you decrease sirtuin activity in yeast and in small mammals, you can decrease lifespan by 50%. If you increase sirtuin activity in yeast and small mammals, you can increase lifespan by 30%. So the sirtuin activity is very important. So, okay, now you have an understanding of the epigenome and the importance of the sirtuins. The second point is sirtuins require a coenzyme, and that coenzyme is called NAD. Without NAD, the sirtuins just don't function. So this is your first glimpse into how these longevity genes are amenable to manipulation. The longevity gene sirtuin needs NAD. So we're going to look at the molecules that activate sirtuins, and those that do activate sirtuins are called sirtuin activator compounds, or STACs. And there are 3 ones that we're going to talk about, and we're going to start with NMN, which increases NAD activity and makes the epigenome more stable. NAD has other functions. It also participates in stimulating PARP, a DNA repair enzyme, as well as ATP production in mitochondria. But we're going to focus really just in on the sirtuin portion. So as we age, our NAD levels decline. And in 2012, it was identified that in skin alone, your NAD level decreases by about 50% between age 40 and 60. So our sirtuin effectiveness declines as our NAD level declines, and thus the genome stability declines, leading to aging. So we want to do anything we can to stop our NAD levels from declining. So because our NAD levels decline with time, we want to replenish them. but we can't replenish them by consuming NAD. NAD is too large a molecule to be taken up into the cell. But we can raise our NAD levels by ingesting NMN. So NMN is the abbreviation for nicotinamide mononucleotide. Now, NMN does enter the cell and is metabolized to NAD. So what's nice about NMN is that it seems to get into all tissues in the body, even the mitochondria inside the cell. So other compounds that can also raise NAD levels don't seem to get into all the tissues, including the mitochondria. And this is the reason why I chose NMN. So oral supplements have been shown to increase NAD levels in all tissues. And the benefits are seen in unicellular organisms and small mammals. Those benefits are improving lifespan, health span, and endurance. In a mouse model, it's been shown that NMN can have a number of health benefits: decreasing vascular stiffness, improving vascular health, restoring fertility, decreasing beta amyloid accumulation in the brain, increasing endurance and exercise capacity. Other articles demonstrate, again, lower associated age, weight gain, enhanced metabolism, promotion of physical activity, improvement of insulin sensitivity and lipid profiles. NMN is also being studied by NASA to decrease the cosmic radiation damage to astronauts while in space, as well as U.S. Special Forces to improve endurance and strength. So if you look at number one, we want to do everything we can to stop epigenetic information from being corrupted. And it appears through unicellular organism studies and studies on small mammals that can be done by maintaining your NAD levels. If you look at number two, it appears that some of the epigenetic information can also be restored that was previously lost. Again, by maintaining your NAD levels, we are able to reset the epigenome back to an earlier biological age. And these two things can already be done, and it can already be measured. I will tell you, I myself had my biologic age measured through DNA testing and found my biological age to be a decade younger than my chronologic age. So this is anything but science fiction. Now, with regards to restoring a complete original epigenome from a backup copy to reverse aging, we're not quite yet there, where we can restore this complete epigenome back to an original epigenome. But that science is fascinating, and it goes back to the work of Yamanaka and Gurdon, which was done 16 and 60 years ago. But I hope that day arrives in my lifetime. Okay, so the second stack on our list is Berberine. And Berberine is also a sirtuin activator, but it also brings in these next two longevity genes, AMPK and mTOR. Berberine increases AMPK or upregulates AMPK, and it downregulates mTOR, which is what we want. So AMPK is an extremely important enzyme. It regulates nutrient supply with energy needs. mTOR regulates the recycling of proteins, or the autophagy, and it also regulates protein synthesis. Now, like NAD, AMPK declines with age. So again, we want to do everything we can to upregulate AMPK. So highlighting these three longevity genes and how Berberine nudges these longevity genes in our favor by increasing NAD and sirtuin effectiveness, also uprating AMPK and downregulating mTOR, you see all these benefits. Facilitating the original DNA code, activates the sirtuins, downregulates mTOR, which is associated with longevity by recycling these misfolded proteins in the cells, increasing your NAD levels. It's also an anti-inflammatory and antioxidant, enhances cellular maintenance mechanisms, reduces senescent cells, facilitates weight loss, maintains brain and cognition, and maintains normal LDL levels. So I'm trying to convey to you how through molecular messaging we can beneficially manipulate the inner workings of your cell in our favor. Now, berberine is found in several natural plants. I don't think anybody should be nervous about berberine. You've probably heard of metformin more than berberine. Metformin's been used in this country since 1995 as a hypoglycemic to treat prediabetes and diabetes. And comparing berberine and metformin side by side, they have a lot of similar qualities. Both lower blood sugar. Berberine has other advantages. And the big one in my mind is that metformin is prescription only, berberine is not. And both are hypoglycemics, and hypoglycemics are being studied as an anti-aging medicine all by themselves. There's an excellent study going on now called the TAME study at Einstein, looking at the benefits of hypoglycemics for the general public. So let's look at the third molecule now, resveratrol. Resveratrol also increases the sirtuin activity and also activates AMPK. Resveratrol is probably the one molecule that you might have heard of. It's that organic compound found in red wine. It's hypothesized to be responsible for the low rates of heart disease in the French population, despite the fact that they have a high percentage of smokers and a high fat diet. Now, yeast is an easy cellular model to use to study resveratrol. 70% of the genes in yeast are common with us in humans. And if you feed resveratrol to yeast, the yeast will live and divide about 30% more. The cellular effects of resveratrol have been shown to translate to real clinical benefits in the lifespan of flies, worms, fish, mice, vertebrae, and small mammals via that sirtuin and AMPK pathway. So this compound is also a little unique in that it has very specific benefits to the cardiovascular system as well as the central nervous system. Resveratrol also functions as a powerful antioxidant. Now, free radicals are unstable atoms that can damage your genome. And again, if you think of your DNA as a zipper, if you unzip the zipper, some of the teeth on the zipper are exposed, and those free radicals damage the teeth on that zipper. So antioxidants help prevent cell damage caused by free radicals. So it's always good having powerful antioxidants in your system all the time. Now, plants naturally make resveratrol to fight epigenetic damage from UV radiation, stress, injury, fungal infections, etc. And red wine has more resveratrol than white wine because the red wines are fermented with the grape skins longer than the white wines. Likewise, wines grow in harsh environments such as Burgundies tend to have even more resveratrol. Now, we don't have a gene that makes resveratrol. Maybe one day we'll evolve to have our own gene that makes this molecule, but right now we don't, but we can benefit from this molecule made from plants. So again, I'm trying to give you this idea of how our longevity genes are amenable through manipulation through this molecular messaging. Okay, so let's leave the epigenome now and get into this topic of the telomeres. The telomere is a protective cap on the end of your DNA. They protect the chromosome ends from being mistaken for broken pieces of DNA that would otherwise be fixed by cellular repair machinery. Just as the metal tips on your shoelace prevent fraying of the tips of your shoelace, the telomere prevents the ends of your DNA from fraying. Now, through a lifetime of mitotic cycles and cell divisions, the telomere gets shorter and shorter with each mitotic cycle. And when the telomere gets short enough, the cell no longer divides and either dies off or becomes a senescent cell. Now, there's an enzyme in your cells called telomerase that is constantly trying to repair or maintain the length of your telomeres by adding nitrogen bases. And Astragalus is felt to act as a stimulant to this enzyme telomerase. So Astragalus longevity benefits actually extend beyond just the telomere. They've also been found to provide maintenance to the immune system, maintenance to the vascular system, the brain, the genome, the epigenome, the bones, the skin, the GI system, as well as function as an antioxidant as well. So a lot of benefits. Again, Astragalus is a plant-based compound. I don't think anybody should be afraid of Astragalus. It's largely found in a plant called Astragalus membranaceus. And again, I'm trying to convey to you through molecular messaging. We can promote benefits through our longevity genes. Some of these overlap, and some of them are unique. Okay, this brings us to our last molecule that we're going to talk about. Senescence is the condition or process of deterioration with age. It's the loss of a cell's power to divide and grow. And wrinkles are a manifestation of mitosis, not creating an identical daughter cell, as there are defects occurring in the epigenome, defects occur in protein synthesis or mitosis because the epigenome is becoming corrupted. And I use wrinkles as an example because you can see them. But this is what's occurring in all tissues of your body. It's the skin, the muscle, the bone, the cartilage, the liver, every tissue, every organ in your body. So if the epigenome falters and the wrong genes are turned on and off, or mitosis falters and we can't create an exact replica daughter cell, the normal cell becomes either a senescent cell or it dies off. And senescent cells just don't take up space. These senescent cells also excrete these inflammatory compounds that affect all the surrounding neighboring cells. So by now you're starting to see how cellular health from an epigenetic perspective is the same thing as tissue health and organ health. This is an example of skeletal muscle, cardiac muscle, and liver tissue. And at varying ages, you see increasing amounts of senescent cells. This is what's occurring in every organ of your body. So senescence is part of aging. Your uniform homogeneous cells become a mosaic of normal cells scattered with senescent cells. And as I said before, these senescent cells just don't take up space. They affect all the surrounding cells as well by that excreting inflammatory material. So this mosaic tissue, whether it's heart, skin, liver, or any organ, just doesn't look as well and it doesn't work as well as this uniform tissue. So there are multiple reasons why senescent cells develop. The DNA is damaged, the epigenome is chaotic, or the mitotic cycle is dysfunctional. And as the human body grows older, tissues accumulate higher concentrations of senescent cells. And yes, we want to eliminate those senescent cells. So there are senolytic compounds that remove senescent cells. And that takes us to our fifth molecule. But this is what we want to do. We want to get rid of those senescent cells that are accumulating in our body. So quercetin is our fifth molecule that we're going to talk about. And it can eliminate senescent cells. So senolytic drugs that take aim at senescent cells have been shown to have significant promise in the anti-aging research. They have been shown to improve health span and quality of life. They have been shown to eliminate senescent cells, and they have been shown to increase the lifespan of small mammals. Quercetin has been most effective in mice when started early, when the senescent cells were just starting to emerge. This is just an example of the many articles and publications that exist in my field of spine and orthopedics, discussing how senolytic drugs can preserve spinal disc health with aging. Research like this is occurring in all subspecialties of medicine with senolytics. So quercetin and all the previous molecules that we've discussed, it's not just a longevity issue, it's a quality of life issue. Quercetin has been shown to maintain tissues and organs, as well as having shown favorable results with the brain, the bones, the joints, the eyes, the lung, the kidney, all tissues. Quercetin may also reverse some of the damage that has occurred from environmental stresses. Quercetin, again, is found in many plants and vegetables. It's in the category of a flavanol or a flavonoid. It's a type of flavonoid. Okay, so this is a summary slide of the synergistic and complementary effects of these molecules and their effects on these longevity genes. And I think this demonstrates that the whole product is more valuable than the sum of the parts, which is again supported in the published literature. NMN is the only one that's not plant-based. It is synthesized. All the other molecules that we've discussed are plant-based found in nature. NMN, by the way, is found in nature. It's just it's produced in the lab. It's synthesized, whereas all the others are derived from plants. So to circle back to where we started, this is not just about lifespan. This is about living healthier, living longer, and having a more athletic life regarding all these categories. There is a common epigenetic ground with regard to maintenance of all these organ systems. There's no silver bullet cream or elixir for any single organism. It's all one unified epigenetic mechanism to maintain all of these organ systems. So the 5 molecules that I've discussed in this lecture, I personally have been taking for years. I've personally witnessed the benefits with my own skin, hair, musculoskeletal system, and cognition. And although it was never my goal to actually put a product together that contains these molecules, I recognized how over the years it was cumbersome to order these and take these molecules on a daily basis, which led me to identify the right sources, doses, and manufacturer to make a combination product of all of these 5 compounds together and make them easily available to the public. I think the science behind these messenger molecules is impressive, and I think the benefits should be extended to all. I think I did a good job explaining the published science over the last 15 years regarding the how and the why. And you will find this information, I hope, compelling. The combination product can be found on my website, Rebesana.com. I would also add that these nature-identical molecules should be taken with food to maximize their response. So the rallying cry, eat right, don't smoke and exercise, has taken us about as far as it can. And I am in no way saying abandon that rallying cry. But the dots today in 2022 are the Sirtuin dots, the AMPK dots, the mTOR dots, the longevity gene dots, and recognizing that they can be nudged in our favor. You now have the basic information to understand how you can slow your aging process to promote a better lifespan, a better health span, nutrition, and fitness, and lead to a more active athletic life. My website, Rebesana.com, also has a lot of articles in the resource section that provide more information, as well as a fairly extensive bibliography if you're interested. So thanks a lot for listening to this presentation. I hope you found it useful and worthwhile. I think if you adopt these principles, you will not look back and regret it. Thank you again.